Today on The Future of Everything, the future of cellular mechanics. Now, we all know that our bodies are made of cells, small living units that have many different types. Our muscle cells can contract and move our body. Brain cells connect to one another and control our thoughts and motions. Blood cells carry oxygen, fight infection. Skin cells provide general protection, covering up all of our insides. Retinal cells in the eye can sense light and send signals to the brain, ultimately leading to vision. Now, each cell typically has a complete copy of our genome, the DNA, and they all derive from a single cell formed after union of an egg and a sperm cell to form a fertilized egg. Now, the last 30 years has seen a revolution in our understanding of how cells differentiate from this single fertilized egg to all the different cellular subtypes. As cells divide, they take on different fates. Some will become brain, some will become skin, etc. And in some ways, the most remarkable part of this process is that the cells not only differentiate into special subtypes, but they also wind up in the right place. They form organs that have the right shape, our hands, our fingers, our feet, our toes, and all the intricate details of a body. How do cells coordinate and communicate during these processes? Hey, I'm supposed to be in the thumb. You're supposed to go over there and become part of the pinky finger. I mean, I don't think the cells talk to each other literally, but they must communicate and they must make decisions about where they're going to go. One of the key questions is the forces and other cues that cells sense and generate for this communication and coordination. This is sometimes called mechanobiology, that is to say, the biology of mechanical interactions between cells, their constituents, and across tissues. Alex Dunn is a professor of chemical engineering at Stanford University and an expert on these matters. Alex, how did you get interested in the mechanical properties of cells and how it influences their behavior? And what are some of the big questions that you're approaching these days?
Well, first of all, thanks for having me on your show today. It's a real pleasure. So I think that the thing that's always interested me ever since I was like a little kid is how do animals build themselves? What accounts for the enormous diversity of beautiful animal forms in the world today? There are a lot of ways of approaching that problem. And the way that I'd like to talk with you about it today is to lead off with a quote from William Faulkner, which is that the past...
Excellent. We have not had a lot of those.
Yeah. Well, according to Faulkner, the past is never dead. It's not even past. And of course, he was talking about the history of the American South, and it's a quote that's actually quite relevant to the situation we find ourselves in today. But it's exceptionally relevant to biology in that you and I carry the history of animal life in every cell in our body, and the particular way in which we have become conscious animals is dictated by the possibly random series of events that led from single-celled organisms living deep underneath glaciers about, say, 800 million years ago to today. Now, you started off with the problem of mechanobiology, and the reason that I wanted to start, let's say, 800 million years ago, is that life had actually existed before that for about 2 billion years. Mm-hmm. And no multicellular organisms. From my standpoint as a, large animal, nothing interesting to talk about. And the big problem was how to build something organized, how to make something that's not just a big glob that has an inside and an outside and a front and a bottom, top, back. And there are two key strategies for that. One is to use diffusing chemical signals that allow the cells that will become your hand to coordinate who becomes a thumb and who becomes a finger. Those are called Morphogen gradients, and I hope that if you haven't already, you'll have a developmental biologist on the show to talk about that.
And just to remind everybody, the idea there is as a cell emits a chemical, there's going to be the areas close to the cell will have a high concentration of that chemical, and as you get further away, so you can actually use that as a measure of distance. And then if another cell can say, oh, I'm seeing a lot of this, then they know that they're near that first cell. But if they say, I'm not seeing very much, then they know that they're further away, and they can use this to do calculations.
Sure. And so I liked that okay when I learned about it in school. I thought it was okay. Like I remember taking classes and the biologists would be totally pumped about how to make sure that a fruit fly had one and only one head. And I'd be like, well, that's good for the fly. I can trust that worked out okay with me. But how do you make something that's finger-shaped or head-shaped or nose-shaped? How do you get shapes and form? That's the question that always bothered me. And in large part, that comes around to the second way that you pattern animals, which is with mechanics. Mm-hmm. The physical forces, physical laws, no vitalism here, no magic, at least of a magical sort.
Right.
And instead, a set of physical rules, some of which we understand and some of which we don't, that transform tens of cells and hundreds of cells and sometimes millions of cells into a defined form. Now we can talk about what some of those rules that I think I understand might be, but the real mystery is, from an engineering standpoint, how do we control those rules? How do we take advantage of the engineering principles that we see around us? And so that's where I spend a lot of my my time inside my head.
Great. So that's thank you very much for that introduction to kind of what drives you. And where are we in understanding? Let's take the hand. The people who might be listening didn't know it, but you were very persuasively looking at your hand just now while you were describing these ideas. What do we know about how fingers are formed? And specifically, how much of it is encoded in the DNA explicitly, where the DNA says, put a cell here, put a cell there, put the bones here, versus how much of it is more of a physical phenomenon with cells that have general rules responding to their environment and kind of self-organizing without explicit instructions. Do we know this yet?
So I think that I think about that all the time, and it's not clear what an instruction is. So to my knowledge, there's no extra embryonic signals that pattern the hand, for example, or for that matter, most of a human, meaning that the instructions to build the shape of a human are contained in your DNA and mine. And we got nutrients and lots of chemical signals from our mothers in utero, but none of those were patterning per se. Now back to the hand. The whole thing builds itself. And it's a very hierarchical structure. The reason I like the hand is because it is so complicated. Lots of little bones, tendons, muscles, nerves, blood vessels. You need everything in order to have a functional hand. Fingernails are probably the only dispensable part on it. No skin, rampant infection, probable death. So the whole thing has to work as a unit. Now, how can we begin to even understand that? Probably it's the work of many hands to understand one hand. So just for example, let's talk about the patterning of blood vessels. That's something that has been studied in great detail and that we kind of understand. So if one of your muscle cells or a cartilage cell or a tendon cell decides it's not getting enough oxygen, it sends out a chemical distress signal, a growth factor called VEGF. And that diffuses through the tissue, and if an endothelial cell, that's a cell that makes a blood vessel, senses it, it sprouts off from an existing blood vessel, begins crawling towards it, and multiplying, and so you gradually get an ever so thin tube extending towards the part of the tissue that needs more blood flow.
Interesting. So it's sort of it's a response to a distress signal.
Yeah.
Let me just say that this is The Future of Everything. I'm Russ Altman. I'm speaking with Professor Alex Dunn right now about the patterning of blood vessels and the response to stress that leads to the first outpouching of a new blood vessel.
Yeah. No. Let's suppose that that blood vessel succeeds in its goal. It reaches a tissue. Well, what good is a dead-end blood vessel, right? You need an outward path and an artery and an inward path and a vein. And so these two will meet, make a capillary bed, exchange blood, and around it goes. But what if it fails to meet a vein? What if there's no return path? Well, then it dies off. Why? Well, because the cells that make up the blood vessel are also sensitive to the physical cue of fluid flow. And if they feel blood flow, then they know that all is well and that they should stabilize and perhaps even grow the blood vessel. And if they don't, then they know that that vessel is non-functional and it'll prune back. So the whole thing is self-organizing. And because you have this cue to grow and cue to prune, you get a system where every part of your tissue is adequately and efficiently supplied with oxygen and nutrients. Yes. I've heard that there's no cell in your body that's more than 2 cells away from blood.
Yeah.
And all of this, not quite all of this, self-organizes. You can totally see this. So you may have heard that retinal scans are a way of identifying a person. The blood vessels in your retina have a different pattern than the blood vessels in mine. We all have one aorta, right? And so I like this system because you can see that some things are deterministic and patterned. How those are patterned is, I think, something that's not well understood. Yeah. Why one aorta and why where it is. But the fine structure is stochastic and emergent, if you will.
Very, very good. So take me into your laboratory. What kinds of systems do you work on that allow you to try to uncover these principles? Take us through a little bit of the day-to-day life of a graduate student or a postdoc working in your lab. What are the systems that they set up? What are the fundamental questions that they're asking?
Okay, so I'm interested in approximately everything. And because of that, there are as many projects in my lab as I can get away with. Now, most of the lab, most of the time, works on really rarefied problems that seem to have nothing to do with anything interesting. And the problem that I particularly like right now is if you have a cell and that cell forms a sheet, just like your skin, those cells will usually define a top and a bottom. So how do they do that? Now that you may seem like a triviality, whatever, top, bottom, they'll figure it out. But it's actually that decision that is, in my view, critical to making any sort of interesting animal. Because as soon as you've formed a sheet of cells that have a top and a bottom, then you can close that sheet and form a tube or a sphere. And you and I are just a series of tubes in some sense.
Yes, yes.
So this decision, this molecular encoding of up and down and self-organization to make it happen is absolutely critical to understanding what an animal is. So,
you have me convinced that this is a critical, especially, and I think this is implied in what you say, when you have a top and a bottom, the top part might have different properties than the bottom part, and therefore you've created like an asymmetry that allows biology to take advantage of one side doing one type of thing and the other side doing another type of thing, and so that you're kind of off to the races.
Yeah, exactly. Now, again, this to you and I, up and down seems like pretty trivial, right? But remember, the thing that makes cell biology hard is that everything is encoded in molecules, inanimate, dead molecules, all self-organizing to give rise to the phenomenon we call life. So this up-down split between cells requires, let's say, on the order of a dozen core proteins and then probably another 3 dozen ancillary proteins to make sure that it works robustly. And they all interact with each other in a complex web. Talking with my hands. They all interact with each other in this very complex web of causal interactions, some of which we understand, some of which we don't understand, and many of which we think we understand but don't. So the problem then that most of my students spend most of their time on is trying to understand how molecules that are too small to see and that are constantly changing in state cooperate to make a cell do this up-down trick.
Yes.
And that's hard because it turns out that these molecules are very small. So you can't just look. Ask yourself, how do you understand the function of a bakery? I, you know,
And a lot of it is looking in the window and figuring out,
Yeah.
inferring how they do things by watching.
yeah, exactly. But do you really know? Right? Probably neither. Speaking personally, I've never been there at 4 or 5 in the morning when I'm told they actually make the bread. So first of all, we can see what's going on, but we usually don't have access to the actual key events. And second, we may think that we understand the important causal events. I walk in and buy bread. But it turns out we understand the core functioning of the bakery only very shallowly.
Yeah, from our own perspective.
Yeah.
This is The Future of Everything. I'm Russ Altman. I'm speaking with Alex Dunn. In addition to speaking about bakeries, we are thinking about the mechanisms by which we can interrogate cellular processes and cellular, really decision-making in some ways.
Mm-hmm.
So you've painted a picture of this fundamental issue of top and bottom that your students address. But I know, and as you said, you're interested in approximately everything. What are some of the other things that the lab is asking about?
Well, one thing that I'm really, really excited about right now is a related problem, which is how do you build a computational machine out of proteins? Now,
So like a biologically based computer.
how does biology build computers? Let's say that.
Okay.
So obviously, there are various forms of biological computation you could appeal to, the way that neurons compute information, the way that information is stored and transformed in DNA. But to me, the analog devices that power our cells are the most fascinating. And what I mean by that is that proteins, in addition to forming the physical stuff of cells, simultaneously take in information from the outside world, including mechanical forces. That's sort of my lab specialty. And then transform that into information that the cell can process. Now, this is a completely different form of information processing than a digital computer. And I, at least, totally don't understand the rules. So in order to tackle that, my lab is trying to push forward the technology that will ultimately allow us to visualize these protein structures in 3D with molecular resolution. So we do quite a bit of technique development around that. And it's early days on that project, but I'm quite excited about the prospects.
This is The Future of Everything. I'm Russ Altman. We'll have more with Professor Alex Dunn about inquiring into how cells communicate and coordinate next on Sirius XM. Welcome back to The Future of Everything. I'm Russ Altman. I'm speaking with Professor Alex Dunn. And in the last segment, we finished up talking about this idea that biology does computations and that the cells in our bodies are doing computations all the time. And in fact, we were then hearing about protein molecules that are the basic building blocks, perhaps, of these biological computers. I want to step back, Alex, and ask you, how should we think about protein molecules? It's not many of us think about Heath bars or whatever, protein bars, and that's relevant, but it's not actually what proteins are. So could you give us a little primer on that, and then we'll move into these computers that are being built out of proteins?
Sure thing. So the really important thing to understand about biology is that biology intrinsically builds from atoms up. So the smallest building blocks in a cell are, let's say, 10 atoms. And then those assemble into structures with, say, 1,000 atoms or 10,000 atoms or 500,000 atoms. And those structures are called proteins. Now, protein is, of course, delicious to eat. As an animal, animals eat protein, and we use it for fuel. But that is the least interesting thing about them. Proteins are a polymer, a string of repeating elements that can fold up into a specific shape. And that specific shape can do all kinds of marvelous things. It can catalyze chemical reactions. It can turn chemical energy into motion and movement. So proteins in your muscle allow us to move, and they're turning chemicals, a chemical called ATP, into directed motion. And they can transform and store information. And all of this is enabled by the fact that you have a long string of repeating units that folds up into a defined shape. So it's like a car that would magically fold itself into a car shape, even if you completely disassembled it and left it out on the pavement. Now, of course, cars are much more complex than a given protein, but you have to forgive proteins in that a typical one is about 5 nanometers across.
So a nanometer, let's put this in some perspective. How big is a cell, or something that somebody might have thought about previously?
Well, another thing that people hear about but probably may not think about is that a typical structure inside of a modern computer chip has features that are, say, 10 nanometers across. So a protein is half of that. But what is a nanometer, right? Well, a meter is, I'm a little less than 2 meters. A nanometer is a billionth of that. Can we grasp what a billion is in our head?
Tough one.
Geopolitics says no. So anyway, let's just say teeny tiny, too small to see.
Good.
And again, we're talking about how you build a machine that's 5 nanometers across out of atoms and have that machine build itself. Then what I find fascinating about cells is those protein machines assemble into bigger machines that can have 100 or 1,000 proteins in them.
Consistent with your earlier claim about the hierarchical nature.
Yeah, exactly. So you and I, when we're building something, we conceive a plan, and then we tend to build downwards. Biology doesn't have that option. Remember, you're starting off with atomic information in DNA, turning that into proteins, which then help to build a cell. So building upwards. And I'm most interested right now in the messiest step in that process, which is how you go from these proteins into giant protein superstructures that help construct a cell. Once you get to this cell, it's still really cool, but it turns out that biology has made the logic behind how cells work together relatively human understandable. This intermediate step, how you build a cell from molecules, is still incredibly difficult for scientists.
And so how do you probe this experimentally? What kinds of things can you do with the modern tools of biology?
Well, it's totally amazing what you can do these days. So there are these incredibly expensive, fancy devices called electron microscopes. And there are a couple about 5 miles from where we sit. They allow you to almost not quite kind of see these proteins inside of living cells. Now, the problem is that even as you're imaging the cell, the electrons, the microscope works by shooting electrons at your sample. And then it measures how those electrons bounce off the sample, and you get information about the structure that way. Now, you and I in our living tissues do not like that. Another name for this is beta irradiation, which is like a not good, bad thing. So even as you're imaging your sample, you're destroying it. And the key is to try to get information about the organization of the cell before you reduce it to its constituent atoms.
Yes. Yes, and so electron microscopy is a key way to look at these. Now, can you see movies, so to speak, of these things, or is it always going to be a static snapshot, and then you have to infer the sequence of events?
You know, that is such a good question. I've always been an enthusiast for structure, for organization, and I was so excited about this in a job interview a long time ago. I was like telling somebody, man, it's everything. And the guy who was interviewing me said it's half of everything. I passed the interview anyway, but he was, of course, absolutely right. He was very kind about it. The dynamics is where the information evinces itself, and the structure describes how it's stored. So my lab has much lower resolution techniques that allows you to watch proteins as they move around inside of cells, single proteins moving inside of living cells. And we're trying to use that to understand the transduction of information.
This is The Future of Everything. I'm Russ Altman. I'm speaking with Alex Dunn. We were getting a feeling for some of the experimental approaches with electron microscopy. And in our last couple of minutes, I wanted to ask, you're in a department of engineering. So in addition to trying to understand the world, and most of our discussion has been understanding proteins, understanding the cells that they create, usually, engineers also have kind of design in mind. And so I wonder, do you have a design element to your work? Are you headed towards creating new capabilities that will be useful for somebody or some activity?
Well,
And where are we in that dynamic trajectory of research?
Well, as Ophelia says in Hamlet, Lord, we know what we are, but not yet what we may become. And
Faulkner, Shakespeare, this is very impressive.
well, if you keep talking to me, I'll run out pretty quick. So humans are at an incredibly interesting point in our history where we're beginning to derive the ability to shape and reshape ourselves. Now, just like everybody else, I'm interested in growing replacement parts, hands, hearts, skin. Skin is harder than it seems. There, I'm told, something like 8 different cell types in a patch of skin. And we work on that, of course. But what I'm really interested in is this. What are the design principles? There's a set of reliable principles that electrical engineers use to design integrated circuits. And if my lab can find just a few of those principles and explain them, I hope that, let's say a couple decades from now, bioengineering will be like other forms of engineering, where you can reliably design the thing that you want. Right now, we totally can't do that.
Yes.
Right now, it's cooking. And so first I'll figure out up and down, as we talked about earlier. Cells also do front and back. I'll work on that one. And then the last thing that I want my lab to be able to do in the next decade is how you make something that is either a hollow tube or a sphere of a specific size. And that will be a useful set of engineering principles.
So size control is a frontier.
Oh, yeah. From an evolutionary perspective, the ability of a class of animals to inhabit multiple sizes and still function is probably a key driver of evolutionary success.
Mm-hmm.
And from your perspective and mine, having 2 hands of very close to the same size is a highly desirable feature. And we actually understand a little bit about how that works. It comes around to mechanobiology. But I see we're almost out of time.
Well, thank you very much. And thank you for listening to The Future of Everything. I'm Russ Altman. If you missed any of this episode, listen anytime on demand with the SiriusXM app.